Alright, welcome back everybody, and today we're going to be talking about Lecture 3. This is part 1 of 2. Remember I mentioned that we are going to be reviewing Lecture 3 twice. I split it up because of the amount of detail. Remember, we're covering 2 chapters here, chapters 4 and 5, both having to do with various parts and different things about the cell. So part 1. So again, reads chapters 4 and 5. Overview. So part 1, we're going to look at the structural difference and similarities between prokaryotic and eukaryotic cells. This course and the coursework you progress through in this program obviously focuses more on the eukaryotic cells, the the animal and human cells. But we will want to know some key differences between prokaryotic and eukaryotic cells. Know the cell membrane and that it acts as a semi-permeable barrier and is made up of a phospholipid bilayer and also has proteins kind of mixed in and amongst that bilayer of the membrane. So we'll look at that as well in some detail. We'll briefly touch on some of the key organelles and their function here as well, but again, part 2, if you've already looked ahead at that PowerPoint and that lecture, which is also posted, not the voice recorded version yet, but you'll see that we focus on the organelles and their specific functions in greater detail in part 2. And then another thing we want to cover here in part 1 is different ways proteins are transported both within and out of this cell. So we're going to be looking at things like various types of transport, such as protein transport, osmosis, the movement of water in and out of the cell, and active and facilitated transport. So again, looking at the differences between the prokaryotic cell and the eukaryotic cell. Obviously, right off the bat, you can tell that the eukaryotic cells are compartmentalized. So they're made up of various organelles and structures and even more membrane-bound structures within the cell. So the cell itself has a membrane, and then within it, the various compartments or the different organelles will also, for many of them, have their own membrane-bound systems. And again, we focus and touch on why that is in part 2. But as you can see here, the other key thing that we have is, again, the many different organelles, whereas with the prokaryotic cell, it's much more basic, much more simple, and you don't have the various compartmentalization, so you do not have the presence of many, if any, organelles. So the nucleus, for example, is just kind of tangled up and kind of hanging out within the cell itself in the prokaryotic cell, whereas in a eukaryotic cell, we know that the DNA is housed and kept specifically in the nucleus. We also have ribosomes, which is common to both. Remember, ribosomes are the machines that actually carry out the translation process. So they take the messenger RNA, which comes from the nucleus, and the ribosomes capture this messenger RNA sequence and convert it into a string of amino acids, which ultimately will form our proteins. And then as we progress, and what we'll see between this lecture and lecture 2, after protein synthesis, the proteins usually, for the most part, will continue and go through the different types of organelles, such as the endoplasmic reticulum, the Golgi apparatus, and then either secreted or kept within the cell. So again, know some of the key differences and similarities between the prokaryotic cell and the eukaryotic cell. So biological membranes. Again, we're going to now kind of turn our focus more towards the eukaryotic cell system. So the biological membranes, right, are barriers that define an aqueous compartment in which the cell internal environment has properties different from the external environment. So what's going on inside the cell, the environment, the acidity, the pH levels, the water, is going to vary slightly compared to, say, what's on the outside of this particular type of cell. Um, Membrane properties depend on the properties of the component parts. So what that means is, you know, if this membrane has more or less phospholipids, for example, um, or does it have a specific type of phospholipid that makes it more or less flexible? And we'll actually see an example of that. So the amount of phospholipids within the membrane, as well as the type of phospholipids within a given membrane, can actually help kind of determine the structure and, in a way, the movement and curvature of a specific cell type. So again, biological membranes are for the most part made up of lipids. Remember, with lipids, basic structure is that they are long hydrocarbon chains. For the most part, they can be saturated, right, or straight chained. Some cases they can be unsaturated or contain one or two double bonds causing kinks. Um, and these are the more prevalent membrane lipids have a glycerol backbone and a fatty acid tail. So again, these are just examples here. So this is palmitate or palmitic acid, again, straight chain. And then at the head group would be a triacylglycerol, for example. So carbon and oxygen. So properties of phospholipids, again, PL means phospholipid. So the phospholipid bilayer will spontaneously close to form a liposome, which again separates what's going on inside of this particular cell formation to what's on the outside. So, for example, this is how our cells, or eukaryotic cells, are. They are types of liposomes. And so basically, remember, with these phospholipids, you will have the uh, polar or hydrophilic head groups here in white, and then the chains or the actual lipid fatty acid tails are going to be hydrophobic, right? So they hate water or want to be away from water as much as possible. So in the presence of phospholipids that contain two tails, for example, they are going to obviously come together, so the tails will attract within each other, and the head groups will face outside or inside of a given environment, because again, they'll be more keen to attract or be used to being in a water or aqueous environment. So again, the phospholipid, or within these phospholipids, which make up the bilayer of a membrane, are going to contain a fatty acid tails, which are hydrophobic, and then the polar head groups, which are hydrophilic, facing the aqueous environments. Okay. So again, this is just a zoomed in or up close version of what this phospholipid bilayer looks like in an actual biological membrane of a cell. This right here is also something to take note of. So the degree of carbon-to-carbon bond saturation determines the fluidity of the core or of the membrane. So again, remember, if the phospholipids that make up a particular cell membrane contain more straight chains and are more saturated with hydrocarbons or carbon-carbon bonds, that means they're going to be more stiff, more rigid, versus something that's unsaturated and that has more, say, double bonds amongst the carbon-carbon connections. And again, just to show an example, so again, palmitate, for example, which we saw a couple slides before, is more single carbon-carbon bonds, is straight chain, so that means this is going to be more stiff and more rigid compared to, say, a cell membrane that contains maybe more oleic acid or oleate here within its membrane, which again has a double bond within its chain of hydrocarbons, so therefore causing a kink, which would then cause this particular cell membrane, again, if it contained more oleate, to be more flexible. And again, this is just another illustration. So, some common phospholipids, for example, that do make up many of our cells, for example, are PC or phosphatidylcholine, and then there is also PE, phosphatidylethanolamin. And as you can see, PC, there is some curvature, there is some kinks within the fatty acid tails, but as you can see, they're also more tightly packed. They're a little bit closer together compared to, say, PE fatty acid tails, which are wider and a little bit looser. So therefore, many biological membranes will vary with their lipid composition. So in this example here, where you have maybe about the same or maybe a little bit more of the PE types, you'll have the curvature or the more flexible portion of the cell membrane. Whereas the phosphatidylcholine or the PC, here in the red cylinders, when they dominate or there's more of them, that's where you have a more straight, rigid area of the membrane. Okay, so continuing on, so not only are there phospholipids and lipids that make up the biological membranes, but remember we also have this thing called a fluid mosaic model. And basically what this says is that there's other things there within the membrane, including proteins. So we have various examples here of different types of proteins found within and around the membrane. So for example, you can have lipid anchored proteins. So these are proteins which then contain a specific shape that will connect or embed itself into the phospholipid of the lipid bilayer here. So that would be a lipid anchored protein. You also then have integral membrane proteins, so proteins that are integrated within the lipid bilayer. So they will have an extrinsic side, so a side that sticks outside of the cell. They'll have an intrinsic side, so a side that sticks within the cell, and then another portion that connects them that is also embedded within the membrane. Okay. And so again, you have both external, intrinsic, peripheral membrane proteins, and then you've got examples of intrinsic or proteins that have a portion of them that are embedded within the lipid bilayer. Now, many of these proteins will have specific properties or shapes about them that allow for this. So, for example, a protein that spans the membrane is usually going to be composed of alpha helices and maybe some beta sheets as well. So remember, the primary structure of proteins is the string of amino acids, which then lead into the folding of the secondary structures, typically alpha helices and beta sheets. Typically what you can do is, as a scientist, you can actually look at a sequence of a specific protein, and if there is a particular portion of the amino acid sequence of a protein that contains a certain group of amino acids, for example, let's say the more hydrophilic types or the more polar, So if we're looking at a string of amino acids that has, say, a lot of serines and threonines, for example, which are polar, um, those might form more alpha helices that have the ability to, say, create a tunnel-like structure that can span the membrane. Okay, so that's an example there. So there is mobility amongst many of these proteins. So some of the proteins that aren't anchored, for example, either to a structure on the outside of the cell or within the cell, um, do have the ability to move around and change position. So some proteins will diffuse freely throughout the membrane, and they move side to side, basically, so they'll move laterally. Other proteins, again, that may be anchored, are going to stand still and stick. They're not going to diffuse freely or at all um, amongst or within the lipid bilayer of the membrane. And so to prove that, there was an experiment done to prove the mobility of proteins within membranes. This experiment is called FRAP, F-R-A-P, fluorescence recovery after photobleaching. So in this particular example and in this experiment, they looked at a cell and they targeted the membrane proteins, and they would dye them with fluorescence labeling. They then take a laser and they bleach out the fluorescence of a particular area or section of proteins. And then basically they simply monitor and watch the cell membrane in a given amount of time. And after some time, what they started to see is the fluorescence was being recovered, or those proteins that were stained with fluorescence were moving in to the areas that was bleached. So in this portion here, for example, which is the fluorescence recovery, what we see is we still see some bleached out proteins. So this tells us that these proteins were probably peripheral, or proteins that maybe were either lipid anchored or anchored to a structure on the outside of the cell. Whereas those proteins that are still stained blue or that have the fluorescence were more mobile, and were those proteins that could diffuse laterally and freely amongst the membrane. So again, fluorescence recovery after photobleaching goes on to show that the cell membrane is in fact composed of a mixture of proteins that can diffuse freely and those that cannot. That's the takeaway on that. Okay. Also, we touched on this briefly. It's definitely touched on in your reading. Is types of cell junctions. So neighboring cells in animal tissue, in particular, often are attached to each other. They interact and they communicate with each other, and they do this through specialized junctions or areas between them that will come together. And so I want you guys to review tight junctions, desmosomes, and gap junctions. And this video, this link to this video in particular, is brilliantly done. It's fairly short, and it hits the point home of specifically what I want you to know. I want you to know basically what is a tight junction and what kind of things would occur there. Gap junctions, for example, those being more hollow connections between two cells, which allow, say, other proteins and ions to flow back and forth between two cells. So I want you to just know the basics. So whatever you see out of that video and from the reading of your textbook on junctions is exactly what I want you to understand. And then, of course, again, key organelles and their functions. So this, again, part 1 briefly touches on that, whereas part 2, I'm going to go into these in greater detail. So the nucleus obviously contains the DNA. DNA is the master plan of and that control the cell's activities, which ultimately is done by directing the synthesis of proteins. Ribosomes are the cellular components that use the instructions from the nucleus, and again, that is written in the form of messenger RNA to build proteins. Again, review the video on protein synthesis. It's very clear and it's fairly direct and easy to understand. So DNA is encoded and is there, the set of instructions within the nucleus. When proteins need to be made, the DNA gets transcribed, right, first into messenger RNA, and then the messenger RNA is exported out of the nucleus, is joined up by the ribosomes, which then translate that ultimately into a string of amino acids, which then fold up and become our actual working proteins. Right? The endoplasmic reticulum, right? We start looking at, again, the compartmentalization, so the different organelle setups. So the endoplasmic reticulum is an extensive network of flattened sacs and tubules and is a biosynthetic workshop. Again, we're going to see what that means and what that is in greater detail. But basically, proteins, many of them will go from being translated within the ribosome and then get brought and pushed through the ER, or the endoplasmic reticulum, where they can get further translated and then further processed or modified. There's two types of ERs, or endoplasmic reticulums. There's the smooth ER and the rough ER. Again, we're going to see the difference between the two in the next lecture, part 2. But remember, rough ER is basically the part of the endoplasmic reticulum that is decorated or studded with ribosomes bound to its membrane. So therefore it has a rough look to it. The Golgi apparatus, again, another membrane-bound organelle within the cell. Many transport vesicles travel to and from the Golgi. The Golgi serves as a molecular warehouse and processing station for products or proteins that were manufactured by the endoplasmic reticulum. So again, what we're going to see is there are proteins that will get translated, they will get filtered into the endoplasmic reticulum, and then they go from the endoplasmic reticulum, they are transported via vesicles, which will pinch off of the ER, get carried to the Golgi apparatus where they are deposited, and typically within the Golgi apparatus what we will see is proteins get modified, and in some cases, many cases, they usually get sugar groups attached to them and become what's known as glycoproteins. And then of course another key and important organelle is the mitochondria. This is where cellular respiration occurs, which basically converts your chemical energy of food into the energy currency used by your body and the cells, ATP. And again, we're also going to have an entire week, still one more week, the 4th week coming up next week, that will be devoted to cellular respiration in great detail. Again, protein synthesis. Can't stress how important this is throughout your careers and understanding of this process with cell biology. So again, review the video. Know that after DNA is transcribed, it goes into messenger RNA, which again leaves the nucleus, ends up in the cytoplasm. Ribosomes will seek out and find or get hold of that messenger RNA, and that is where the translation process occurs. Some ribosomes are free in the cytoplasm, others are attached to the ER. And remember, if ribosomes are attached to or bound to the endoplasmic reticulum, then that portion of the ER is known as rough ER. Okay, and then protein export. So this is basically where proteins, once they are processed through the endoplasmic reticulum, they will usually either be secreted out of the cell completely, or they go through the Golgi apparatus for further modification, and then they get released or secreted out of the cell. An example is insulin. Okay. Um, so how does this happen sometimes? So a polypeptide chain, so a chain of amino acids, as it's being translated out in, say, the cytoplasm with the ribosome, what will happen, and again we're going to look at this process in great detail, the mechanism itself in part 2, but basically what will happen is a ribosome, which is either already bound to the ER or which will become bound to the ER, finishes the translation of the protein, and as the protein is being made, that protein or that polypeptide chain is fed into the ER. And then once in the ER, a lot of times some processing will occur. So short chains of sugar are then attached and linked. And then again, when ready for export from the endoplasmic reticulum, that protein that has gone under further modification will get packaged into a vesicle. That vesicle buds from the endoplasmic reticulum and gets taken typically to, say, the Golgi apparatus. And then that protein again undergoes further modification. Sometimes it'll get more sugar groups added to it. Other times it might get trimmed and lose some amino acids or other features. Then that protein is then finally transported again through a vesicle from the Golgi. That vesicle is then taken to the plasma membrane of the cell, where it is then ultimately released out of the cell. So for example, with insulin, in the final stages, it'll go from the Golgi, where it's processed, and once it's proper fully formed insulin, another transport vesicle will take it, bind it to the plasma membrane, and release the insulin out into the bloodstream. Remember also the fact that we mentioned at the beginning of this lecture that the membranes, or the plasma membrane, are permeable or semi-permeable. So this is a list and an example of molecules that are permeable or slightly permeable. So this is an example here and a list of some examples of molecules that can pass through this cell membrane. I definitely want you don't have to worry about the permeability coefficients and understand that number, but what I do want you to know is some examples. So nitric oxide, for example, NO, carbon dioxide, nitrogen, oxygen, these are gases that are permeable. That means they can pass through the cell membrane without any help. So they can go through based off of concentration gradients, even, for example. Small uncharged polar molecules are also permeable. So ethanol, right, or as we all know it as alcohol, can pass through the cell membrane quite easily without any necessary help. And then you have other things such as urea or even water, which is slightly permeable. So that means that they might require the assistance of an active transporter or a protein channel. So water, in some cases, can diffuse through cell membranes via osmosis, or it might require a protein channel that's embedded within the membrane. These water channels are also known as aquaporins, for example. And then other things that cannot get through the cell membrane, at least not on their own, would be glucose, for example, fructose. So these are monosaccharides, as we know them, right? These simple carbs. Other things too, like calcium, chlorine, magnesium, ATP, and whatnot, cannot get through, at least not on their own. But so these are examples. So these things that are impermeable to the cell membrane will require the assistance of protein transporters. And again, we are going to see examples of these in just a moment. So again, osmosis is the movement of water across a selectively permeable membrane. Tonicity is something that you need to understand too. So that's the ability of a surrounding solution to cause a cell to either gain or lose water. So be familiar with a hypertonic solution is compared to a hypotonic solution. So protein transporters, okay? So again, these are going to be proteins that are going to be embedded within the membrane, for example. So these would be uh, integral membrane proteins. And what they do is obviously they're made up of amino acids, they are proteins, they are part of the cell membrane, and they will help molecules that cannot on their own get in and out of the cell. They will help get them in and out of the cell. So these are just examples, and again, we're going to look at some in detail. So, for example, there you have here as an example, and we'll see one, is an ATP powered pump. So ATP, remember, is adenosine triphosphate. That is the energy currency of the cell and of your body. So ATP is utilized here as a source of energy to help move a molecule of some sort in and out of a cell. Now typically you're going to need energy to move a molecule against the concentration gradient. So think of it like this. Think of it as, jamming your fist into a tight jar, okay? It's not easy, but with enough force, with enough energy, you might be able to shove your fist into a small jar, okay? But it requires energy, it requires force. That energy comes in the assistance of ATP, okay? So again, taking something from, say, an area of already low concentration and putting it into a space of already high concentration. That requires work, that requires assistance, it requires energy. You're going to use an ATP-powered pump. Other examples, it would be a uniporter. So a uniporter is simply a protein channel or an embedded protein within the membrane, and it will basically help to transport a molecule from an area of high concentration to low concentration. So help assist it go down the concentration gradient. We're going to see this example with glucose. So remember, glucose in the slide prior here is not able to go through a cell membrane on its own. So what we will see is glucose molecules can still get into a cell via a uniporter or a protein called GLUT1, G-L-U-T-1, and we will see that mechanism in just a moment. Okay. And then other times you'll have symporters, which is where two molecules will go through the same protein channel, but one of them is going against the concentration gradient. So one molecule could be going from an area of low concentration to an area of high concentration, and in that case it will require help or assistance, and it gets that help or assistance with the other molecule that is also going through that same protein channel. But the opposing molecule is going from an area of high concentration to low concentration. And because of that being much easier, it provides energy and the ability to assist the other molecule, which is going against its concentration gradient. So, in a symporter, one molecule goes against the gradient, the other one goes down with its gradient. So here's an example of a uniporter system. So we have glucose, and basically, remember, glucose we need, right? We need to bring in glucose into our muscle cells for storage in the form of glycogen. We need glucose to come into our other cells to provide us with energy to run or walk or carry out the day. So what happens is when you down, say, a Red Bull as you're studying and cramming for this course, you are getting full of glucose. Lots of sugar is going through your blood. Now, obviously, the glucose needs to get into your cells to keep you moving and keep you motivated. So what will happen is glucose will hook up with a transporter. And this transporter is a protein called GLUT1, G-L-U-T, number 1. And what happens is that this protein, the GLUT1 transporter, which again is embedded and spans the cell membrane of a cell, let's say a muscle cell, what will happen is the glucose molecule, as it's flowing on the outside of the cell, say in the bloodstream, will bind to the GLUT1 transporter. Upon binding, this GLUT1 transporter undergoes a shape change or a conformational change, and now it faces inward, so it opens up on the inside of the cell, thus releasing the glucose. And then once the glucose molecule has been released, say into this muscle cell, then it goes back to its original conformation. And as we can see based off this triangle here, we are showing that glucose is going from an area of high concentration to an area of low concentration. So this is also known as facilitated transport, because this GLUT1 transporter is simply assisting the transport of glucose down its concentration gradient. So again, you drink a Gatorade, you drink a Red Bull, that energy, that glucose is flowing through your bloodstream. It's got to get into your cells eventually. This is how it does it. The glucose in high concentration, as soon as you drink that soda, Red Bull, or Gatorade, binds to the GLUT1 transporter, which is always facing outward, to capture the glucose molecule. You undergo a conformational change. This then allows you to release the glucose inside the cell, and then it goes back to its original conformation. Okay. So that's a uniport system. That's also an example of facilitated transports. Then we have an example here of an active transport. So there are pumps, or a particular class of transport pumps called P-class pumps. And these P-class pumps use ATP. So remember, ATP is the energy currency of the cell, and it basically helps to move things, again, also in and out of the cell. Now, usually what you're trying to do, if you're using ATP, remember, you are actively transporting something, and you're usually going against the concentration gradient. So you're usually taking, say, a molecule of, say, calcium, for example, this Ca2+, which is going to be our example coming up, and basically we want to take, for example, calcium, and we want to take it from an area of low concentration and put it into an area of high concentration. So we basically want to force a little bit of something into an area where there's already a lot of it to begin with. And we do that through the use of an ATP pump or a P-class pump. So this is how the calcium ATPase mechanism works. So again, we have this setup here. So our pump here is this entire, again, protein that spans or goes across the membrane. Now what we're looking at here, for example, is a muscle cell. So the muscle cell has a space called the SR lumen, or the sarcoplasmic reticulum. And this is a storage area where within muscle cells where calcium ions are stored. And so within the sarcoplasmic reticulum, you have a high concentration, a high volume of calcium. But many times what we are going to do is take calcium from within the cell and we want to put it into the sarcoplasmic reticulum, or the SR space, here the SR lumen. Now again, because we're going to take some calcium, which is only a little bit within the cell, and force it into an area where there's already a lot of it, we need energy. So this is what happens. So calcium ions initially will bind to two areas or two spots here called affinity sites within this ATPase pump. So two calcium ions bind to the affinity sites here of this pump. This, at the same time, we have ATP binding to another area of the pump. The ATP, when it binds to this other part of the pump, a phosphate group from ATP, which is adenosine triphosphate, one of those phosphates will move over and bind to a D residue. Remember, D is an amino acid, and it is the aspartate or aspartic acid residue. So again, two calcium ions bind first to high affinity sites for calcium. ATP binds to another part of the pump, and then here a phosphate is taken from ATP and binds to an aspartic acid residue on this ATPase pump. Now when the phosphate group moves over to the aspartate residue, it's going to cause a conformational or shape change. And what happens is basically the part of the pump that was closed off is going to open up and allow the release of the calcium ions. And once the calcium ions leave, the phosphate that was on that aspartic acid residue also falls off. And then the P-class pump goes into another conformational change and goes back to the original position. Okay? So again, the takeaway from this is that this is an example of an active transport which requires energy. We are moving something we are taking molecules from an area of low concentration and putting it into an area of high concentration. Okay? Think of it as cleaning up your house right before your mother walks in the door. You are grabbing every piece of clothing off the floor and you are shoving it into your already full closet. Perfect example right there. Okay? But that requires energy, as does moving calcium from the cytosol to the SR lumen. So, calcium binds to the pump, ATP binds to another part of the pump, a phosphate is transferred from ATP to an aspartic acid residue, causing a shape change. This shape change allows the release of calcium, followed by the loss of that phosphate group off of the aspartic residue, and you go back to the original conformation. That's the takeaway I want you to get from this. And then one of the last things that we'll look at here in part 1 are enzymes. Remember, enzymes are also made up of proteins. They speed up the cell's chemical reaction by lowering the energy barriers. Energy must be absorbed to alter bonds in reactant molecules. This is done through the activation energy. So remember, the activation energy is the amount of energy needed for a reactant molecules to move uphill and then downhill so a reaction can begin. So remember, reactants. So for example, carbon dioxide and water are your reactants. We want to form this specific product here, H2CO2. So without an enzyme, it's going to happen, but it's going to take a long time. But if we throw in an enzyme, remember, the enzyme will lower the activation energy and help this reactants come together and form our final product much quicker. So again, enzymes are catalysts. Okay? And this is basic mechanism of how an enzyme works. So an enzyme kind of sits in a specific shape that allows for the binding of the substrate or your reactants to come into what's known as the active site. So the active site is where your substrate or your product or your reactants bind. This causes the enzyme to go through a conformational change, thus bringing together and tightening up and bringing your substrates or your reactants closer together, forcing them to react. And then the enzyme will go through another shape change or conformational change, and now your new products are then released. So that's a basic overview of the enzyme. And then with enzymes, there are optimal conditions. Shape is obviously central to function. So if the shape of an enzyme is altered and substrates cannot bind, then that reaction will not occur. Temperature, which affects molecular motion, can either help speed up an enzyme reaction, or if you exceed the temperature, you can cause proteins to denature, which could cause the enzyme to lose its shape and its ability to do its job properly. And just as temperature, pH levels. So how acidic or basic an environment is for an enzyme can also either help it and enhance the enzyme's ability, or it can hinder and prevent the enzyme from doing its job. And then the different types of inhibitors which affect enzyme activity. So a competitive inhibitor reduces the enzyme activity by blocking substrate molecules from entering the active site. So a lot of drugs, for example, that are produced in the pharmaceutical industry will do that, right? So there are drugs that people can take, say for pain management, and what those drugs will do is they will mimic or be shaped like the standard substrate, and they basically will bind to the enzyme, and then they'll prevent the original or the proper substrate from interacting with the active site of the enzyme. Okay? So again, competitive inhibitor, you're going to basically block the substrate molecules from entering the active site. Then in a non-competitive inhibitor, that drug, for example, doesn't necessarily bind to the active site, but it might bind to another part of the enzyme, and again causing it to change shape. And if you change the enzyme's shape, then it will also not be able to bind to its proper original substrate, thus causing it to be inactive. And that concludes Lecture 3, Part 1. Again, a brief overview of some key features about the cell and things that go on within the cell. Lecture 2, we're going to look at the organelles in great detail and their functions.